Good evening. Today I'll cover basic terminology as well as I'll try to motivate or encourage your question word. Question word. What question word do you know from English? Question nouns and nice. Which? Sava said which and when. Yes. When. How. What. Oh, very nice. What?
And why?
Huh?
Why?
Why? Yes, very nice. Why?
Who and where?
Who? Oh. Who?
Where?
Where? Then let's count. 1, 2, 3, 4, 5, 6, 7 total. Among these, I put why, how, which, right? All three are unique relative to the other four. I'll explain why. Do you know the difference between these three and the other four? Who, where, what, when, against why, how, which. This is quiz. Quiz in English.
I think these are more logical than those four.
This is kind of open question, so we do not have correct or no. So you are saying why, how, which are more logical word than the other four. Then how do you say about the other four?
I mean their answer will be more logical and reasoning.
How
These are
about
quite simple one.
your question, Saba or Mui? One question is why, how, which are more logical than the other four? If I add something more, where? More about space. When? Time. Question about time. What? About thing. And this is about person. Right?
Yes.
I told you these are about English, a question in English, like those are questions in crystallography. So if you have any idea, it will be fine.
We have to explain in, like, there's no one word answer to them. We have to give explanation, maybe.
Yes, this requires explanation, lengthier or more explanation than the other three. For the other four, these requires simple question like who did, then Muhammad or Sabah or Totimui or Tessang. But where, like at crib or at UST, simple question can be answered. What, where, when, likewise. But my version of this is here. I have the other word like this: who, ever, wherever, whatever, whenever. We can put ever there, but these three don't have such a ever word, except how.
Yeah.
But what is the meaning of however?
Very different.
Very different from how.
Question.
So this is my version of explanation. Muhammad's version is logical and more like physical, if I add something like that. Physical versus logical. So Saba's version is simple question can be answered here and more complex we need. So at the end of this classroom coursework, you will do more complex one than this one. Why crystallography research is important and how such a research is possible and which data we need for such important research. Okay. After this one, you will go through your memory and your significance, and you will write down what you studied. Okay. It's kind of a bonus answer. Did you ever think about ever question in this one?
Ever question?
I said ever. Those three don't have ever.
Yeah.
Yes?
Yes.
Oh, yes. Interesting. You are very
we
lively.
observe but never think like that.
But anyway, if I add something more, in English word we have know how. Have you ever heard of know how?
Yeah.
But the time we use, this was used earlier, but recently we are talking about know why. But not yet I did see know which.
Yeah.
But how is about more about technology. Why is more about science. And which is more about culture or value system. Okay. But I will not talk about that. You may develop your own version of such a vocabulary.
Mm-hmm.
Last time we talked about crystallography as a language, and I introduced you a new dimension in your thinking, and that's the point I'd like to share with you from some linguistic point of view for your study. In such a you can deepen your language perspective of physical research like crystallography. Then learning crystallography or studying crystallography will not be boring anymore, particularly for Muhammad. Okay. And please keep those perspective. And now electron density and structure factor. Thought we may be new to electron density and structure factor, but don't be surprised. Just these are key idea about crystallography. On top of your interest in proteins, we are studying crystallography as a way to study protein structure. So beyond this, we will not introduce any more mathematics. No more mathematics. But even here, we will approach these two main ideas through the language, English language, not mathematical language. That is the point I shared with you last week. So I took two ideas or two explanations from wiki. Time to time I found that wiki contained very good encyclopedic description, but still we have difficulty to adapt such an explanation to our specialty. So here electron density is measured. I'm just reading the sentence from wiki. Electron density is measure of probability of electron being present at a specific location. But what is a specific location here? If I translate such a specific location into symbols used in the textbook, then X, Y, Z. Here, X, Y, Z. In Wikipedia, X, Y, Z was never explained. So I put X, Y, Z as a key symbols upon key idea of electron density. With the word electron density in English, I'm adding X, Y, Z as a symbol. But you will not need to study mathematics behind electron density. You are just required to decode X, Y, Z coupled with electron density symbol rho, Greek word rho. That's all you need. No more. No more. No more is very important. Also, structure factor in terms of symbol, F HKL. Remember, X, Y, Z are English alphabet, right? HKL are English alphabet too. F is English alphabet in capital. But what is this? Mui. It can be P in English, but this is not English word. This is Greek alphabet. The other two know the answer, so I'm just asking you. But if you are careful about listening, I already pronounced this. I said ro. Ro is RHO. Then in Greek, written like this, ro. So you need to extend your vocabulary even in Greek alphabet, because we will use alpha, beta, gamma. We already have something here. What is this? Alpha in Greek letter. Yes, Greek alphabet. Alpha. Likewise, rho, alpha, we have beta and gamma too. Beta and gamma, we will use them in geometry session, crystal geometry. Okay? But read such a sentence from Google, not Google, Wiki, and try to translate what you can find from there to the symbols used in textbook. Then you may develop kind of dictionary. So what I'm trying to help you is you need to have proper dictionary when you study something. When you study English for the first time, you relied on your mother tongue to English dictionary and English to your mother tongue dictionary. Which one did you use, Sabah? Or you had knowledge about English when you were very young and you didn't need any dictionary?
Yeah, for like advanced words, I use dictionary from English to English. Like
you used English
to
dictionary.
Urdu, yeah.
But you still need English Urdu
Yeah,
dictionary.
sometimes.
Sometimes.
Yeah.
But how about Urdu and English dictionary?
I never.
Never. How about you, Muhammad?
We never use Urdu to English dictionary.
You use the English dictionary too?
Yes, English to Urdu dictionary. It was used for the specifically when we read the English language, not in the science. Science
Literature. You need more vocabulary than science.
In science, mostly we use just English to English dictionary.
For science, you learned science through English, not through Urdu. So you didn't need such a bilingual dictionary.
Even some terminology in Urdu, for the scientific terminology, that will be difficult to understand. In English is more easy.
So you had most scientific vocabulary already in English, never in Urdu, through your education. But how about you in Vietnam?
Oh yeah, the first time when I started learning English, I used Vietnamese and English dictionary, and then after that, and now I use the English dictionary. Yeah, actually I never ever used English and I did English dictionary for science. Almost only reading the book, English science book, and I got the vocabulary from book.
So you learned science through English, so you didn't need very much about English, Vietnam English dictionary in science.
Yes.
It's an advantage. But even for English dictionary, in terms of definition and keyword, if you use more difficult word in explanation or definition, it can be hard to understand. So we should use simple or easy language in the definition or terminology. rather than keyword. Likewise, through this coursework, I'm encouraging you to develop your own dictionary or vocabulary and explanation. Your version, not the textbook version. So through the coursework, I'd like to help you. Unless you understand some idea, as I explained earlier or last time, we will just cover few ideas. Likewise, here, electron density, structure factor, that's all. How they are related and why they are important and which one we need at the beginning and which one we need for analysis. So, equipped with 3 question words, why, which, how, you will play with these 2 main ideas, electron density and structure factor. But to describe electron density, we need coordinate x, y, z. To describe structure factor, we need coordinate HKL. But if I add something more, x, y, z in fractional term, HKL in integral term, we will explain later why x, y, z are called as a fractional and why HKL are called integral. Any question? Do you know why? Muhammad? It's okay not to remember anything. Anyway, we will cover later. So right now, just be familiar with this rho as a symbol for electron density. Either we can use full English word electron density or simple single letter rho. Likewise, structure factor as a full word, F as a single symbol. That's all. F stands for structure factor as a symbol. Rho, Greek letter rho, stands for electron density as a symbol. Okay? Then, then Fourier transform. This is the answer how these 2 are related. Electron density and structure factor are related through Fourier transformation. Have you ever heard about Fourier transform in mathematics or technology or engineering? No? Then you can safely ignore Fourier transform as a way to do some computation. But as an idea, you still need Fourier transform as a vocabulary, also as an idea. I said, you do not need anything about Fourier transform as a mathematical computation, but you still need to understand Fourier transform as an idea and vocabulary. Vocabulary. I'm not sure about spelling. Spelling correct or wrong? Which spell is wrong? L? L? Not R? Then my pronunciation may be wrong either. Vocabulary. Vocabulary is an idea, not this is wrong. Idea and vocabulary. Fourier transform as an idea and vocabulary. So just take those this word Fourier transform as an idea to explain what. In previous slide, we covered 2 ideas.
Structure factor into the electron density.
Electron density and structure factor should be related by Fourier transform. Okay? So just take this word to explain such a magical relation between those 2. Okay? So just take Fourier transform as your what is magic word? Spell. magical spell. Okay. Spell to connect electron density and oh, good structure factor. I'd like to hear promptly like structure factor from you, and that's all. No more. Be sure we know two functions: electron density function and structure factor function, or two different kinds of data: electron density data or XYZ coordinate. I said electron density and XYZ coordinate data. And structure factor or intensity, HKL intensity. I'm adding more vocabulary. You may notice what I said previously and now. But by reading the textbook, you can develop your own vocabulary at your own pace. Okay? So don't be surprised at all. But here, if I add some more, this is not about crystallography, but general science or engineering disciplines. You are studying biology, but biology is relatively free from mathematical hardcore science or engineering practice. But most engineering and science, hardcore physics-based science, are dealing with mathematics. So Fourier transform is so central in signal processing and information and telecommunication. There, unlike Fourier transform in crystallography, they are using Fourier transform in different coordinate system, so-called domain. Here, you can found two domains.
Time domain.
And what else?
Frequency domain.
Oh, nice. Time domain and frequency domain. Both connected. Time let's say frequency. Frequency f as defined by time is like this.
Inverse relation.
Yes, inverse relation. Yes. Or in terms of number, this is called so-called reciprocal. Likewise, this is in time. T in time, period in time. This is so-called period in time. So usually time itself we are writing T, but period can be written as capital T. And if we have some temporal occurrence, if you have repeating pattern in T, this is so-called T. As a signal, this way, this is coordinate of time. 1 second, 2 seconds, 3 seconds, 4 seconds, likewise. Then T in time is equal to 1 second, right? Periodic occurrence in time. I said periodic occurrence in time, periodic event in time, or periodic happening in time. So if we have signal like this, we can deal with Fourier transform, and we can have frequency, single frequency, 1 second. Actually, corresponding frequency is 1 hertz. Is 1 second. So mostly in engineering, they are dealing with communication over time. So time domain, frequency domain. But in crystallography, we are not dealing with time. What dimensions are we dealing with in crystallography? Yeah, electron density are in time or something else? Oh, good, very nice. Space. This is telecommunication. This is in telecommunication. But in crystallography, we are dealing with signal in space. Okay? They are in time. Telecommunication in time. Crystallography in space. Okay? So, although Wikipedia just described Fourier transform in time domain and its reciprocal dimension in frequency domain, crystallography, we are dealing with space, not time. So for space, we have, so frequency can be called as a reciprocal time, likewise, but reciprocal time is never used in formal telecommunication terminology, but I just add reciprocal time as a short word for frequency, or longer word for frequency, not short. Short was as a single word frequency, but frequency by definition is reciprocal period, reciprocal t, okay, or reciprocal time dimension. But we have space in crystallography as x, y, z, and we have reciprocal space. We will introduce reciprocal space later, but as a symmetry in terminology, you can develop such a vocabulary in advance. Okay, does it make sense? Reciprocal space, like a period and frequency domain, we have space domain and reciprocal space domain. That's the symbol used here, x, y, z corresponding HKL. HKL in reciprocal space, and x, y, z in space, and space domain and reciprocal space domain, like time domain and frequency domain. That's all you need. Likewise, here reciprocity or inverse relation, we have such an inverse relation also in space and reciprocal space in crystallography. Okay. In terms of question, I already covered how crystallographic research is possible. Either you mentioned or not, here I took not from Wiki, but from International Union of Crystallography, so-called IUCR. IUC in capital and R as a small letter, but as a web pages, IUCR in all small letters. Education pamphlet, from that pages I found introduction to the calculation of structure factors. But if we take inverse form, based on structure factors, we can calculate electron density. They are equivalent in terms of information. That will I cover. But anyway, how crystallography research is possible? You can find you should use Fourier transform as a relation between these two different data, electron density data and structure factor data. But can we observe electron density data directly from crystallographic observation?
No, we
No.
need first structure factor intensity, and then we will convert it.
Why no? Let's think about later. He has some prior knowledge, but the other two may not know why not. We will talk about why crystallographic research is important with these two. And I'll cover more later. This from textbook. Have you ever visited protein data bank web pages? Muhammad, yes. How about Toti Mui?
I saw it from the paper.
Paper,
Some paper.
from some scientific paper. How about you, Saba?
Just randomly, not like I didn't thoroughly
Mm.
checked it.
You didn't check it for yourself.
Yeah.
You just heard about PDB or such a data bank.
Yeah.
It's quite normal. It's okay. But when you read textbook, you have too much information. Protein data bank already in this year we have. I captured something here. At the end of year 2008, we had 50,000. Now we have 100,000, over 100,000. So human gene total, how many human genes do we have after human genomic project? You know, total number of human genes, human genome. I'm checking your basic understanding. Human genome as a total contains 30,000 genes. Now we have more than the number of human genome in terms of protein expressed. But here we have redundancy. Not only from human, we have many from bacteria, even plant. So we have different species, even synthetic one. So we cannot say we have more than the number of human genome proteins, but we can say we have now enough proteins. If you are talking about a unique protein folding structure, now we approach the limit, no more new folding. You know protein folding?
I know.
What folding do we have as a secondary structure?
Usually it's a second step.
Secondary structure. Primary structure is a sequence. What is secondary structure of protein?
Alpha and beta.
Yes, alpha, beta, and non-ordered region and coil. And on top of that, we have some quaternary structure, and by combining such a quaternary structure, we have some unique folding, connectivity between protein sequences or peptide. So now we are approaching kind of saturation in terms of protein folding, but we didn't reach at the level of genome level. Even for human proteins, we do not yet have a structure from, we have some, but we do not have all for membrane bound or simply proteins of which structure crystals cannot be made. So we have some lack of coverage there, but anyway, we have some exponential growth in terms of number of deposit structure here as explained in this equation. But interestingly, I discovered something wrong in that equation, and I'll explain later as your critical reading. Any protein, if you have protein structures, question? What advantages or how they can be beneficial for mankind?
Protein data bank.
Yes. Why, as a figure number 1, the author of textbook presented this figure as the importance of crystallography?
This is just as a record, to keep the record of knowledge, so that the next researchers or scientists, they can utilize the previous one knowledge. I think that's why it is increasing exponential rate.
They show different structures of different proteins. So like we can know their properties and their functions by that. So just like this antibody, how can it work? How does the
is important to know the structure of protein.
Art is saying I made this question in wrong way, but you gave me good answer, better answer. As emphasized here, at the beginning of crystallography, myoglobin, hemoglobin are only two at the time of opening of such a database. Myoglobin, hemoglobin are from blood of a horse, and they were not made by genetic engineering. They were taken from natural source. And we just discovered how these enzymes, we cannot say enzyme, is that enzyme or not?
No.
Kind of catalytic activity to transfer oxygen there, right? Is that right, the description? Then lysozyme is enzyme.
Carriers.
Carrier. Oh, better word, carrier. Protein carrier to deliver oxygen to the cell. Right? And as you said, antibody and viral structure and some structural protein like actin and even ribosome for protein synthesis. So through the studies done with protein crystallography, we are discovering more structural aspect of biology, so-called structural biology. So with the advancement of protein data bank, we have observed deeper or broader understanding of structural aspect of biological function, right? That is my paraphrasing your answer. But why I said I made a clumsy question is like this. Actually, at one point, he presented this figure as an introduction is like this. Among these numbers, as a percentage of contribution, X-ray is over 85%, almost 90%. What else were done with NMR and minor technology like electron microscope and neutron diffraction. But still, we have major contribution from X-ray crystallography for structural determination of protein. And upon that, as you explained, variety of structures were understood or discovered from structural perspective. That is good answer against my bad question. So this is about why crystallography is important in terms of understanding, better understanding of biology in functional term by having variety of proteins, even photosynthetic center. Okay. Then, what do you have in your mind with this figure?
Here's a process from beginning of gene cloning from DNA and then transfer into protein, and then with the final purpose, we want to detect the structure and the function of protein.
Then let me ask this way. I may have a poor question, but here we have funnel. This is funnel, right? But what is this? Triangle. But why here and there we have different amount? And can you explain from that perspective? Funnel versus triangle. Then let me ask another question like this. What is the meaning of this attrition in English word? I'd like to check your English capability. Honestly saying, when we read a textbook, we have tendency to skip some vocabulary when we have difficulty to decode it or understand the meaning. But normally it's safe when we have some main idea. But it's not safe when we cannot get the point. Here, I'd like to check the point. If you miss this, you cannot catch meaning here. But I found attrition as an English word may not be easy for some of you. So I'm asking this question. What is simple word or easier vocabulary for attrition? You may find corresponding or equivalent word here. Huh? Oh, nice. So given the high failure rate, you took here? You guess. So failure rate. Okay.
The failure is increasing, and the cost of failure is increasing as the procedure will continue. If we fail at early stage, the failure cost is less.
Oh, yes.
If we fail at the late stage, at the end stage, all the work done is going to zero and the failure cost is more.
Yes. So total budget for project to determine at least one protein structure may be around cost like this, 50,000 or over 100,000. Okay. So, but primer design costs like that, right? So it's better to try more at early stages than having failure at later stages. This may include salary for one researcher, yearly salary for one researcher, and some purification step and some material for cloning and purification, etc. Even considering your time, if you fail at the end of PhD defense, it can be very fatal. But if you fail at first year or first semester, no damage. So it's better to design comprehensively and make sure that you can have enough number of success chance at the later stages. So anyway, we have cheap failure at the beginning and we can have expensive failure at the end. So this is to give you an idea about cost aspect, either in terms of time or in terms of money or effort. So we started with question of why, how, which. But we also have question of like who, where, what, and when. So not only as a scientific discipline, when you plan in research in protein crystallography, we do not have equal demand at each step, so we should manage the project accordingly. So we have many perspectives, not only scientific perspective, but also management perspective. So you should not act as a student, even though you are PhD candidate or master's candidate. You should act as a manager of project. Otherwise, you may be in failure at the end. So this is about learning such a management aspect of protein crystallography. So like drug discovery, this is very parallel to this drug discovery. At the end of drug discovery pipeline, we do not have very expensive failure. We just have very cheap failure. But very at the end of pipeline, like a clinical trial, with single target or several few targets, we should do patient screening. So it's a human environment. So after failure, it can be very costly failure. So likewise, protein crystallography has some high success or high failure rate at the beginning and at the end. So you should be careful about designing your experiment. This is to give you an idea. Now, overall process. By now, you can ignore phasing, but phasing has something with Fourier transform. But except phasing, what do you have in terms of understanding, or what difficulty do you have in terms of understanding this overflow, except phasing or data? Or what topic or stages are familiar to you? Simply let me ask a question. Do you have experience in cloning, Saba?
No experience, but I have seen the work of Muhammad.
Colleagues are working on cloning, Muhammad and other.
Yeah.
Gentlemen. You have cloning,
Cloning, protein expression,
but not crystallization, no data collection. So cloning and expression.
Yeah.
Okay. But what do you have?
How to crystallization?
Crystallization.
Yes.
Crystal.
Yeah.
Out of protein purification.
Protein purification.
Protein purification, this is SDS gel learning for purity confirmation. Then once we have 99% or 95% purity, we can proceed crystallization. We have several kinds of crystallization, but all of them are based on difference in solubility. Even during purification, you have solubility problem or solubility test, like expression test. Then if you have precipitation, you can play with the speed of such a precipitation. Then without having aggregate, you can have beautiful crystal like this. So out of soluble protein, you will have crystal when you play with solubility, with precipitating solution. Soluble protein means your proteins in solution, not in aggregation. But if you add something as an extra to control solubility, then you will have crystal. So you should add some precipitating agent there for crystallization.
And we got the data. Data is the picture or there's some.
Oh, very good question. Here, picture.
Yeah.
But to do computation in crystal, we need numbers. Picture-like data shown in computer display, but we should reduce those pictures or images into numbers stored in computer file. So we should convert image into numbers, so-called data. Data in FHKL, structure vector data or intensity data. Okay. Very good question. But as you told me so, here in this semester, we will cover gene, soluble protein, and crystal, and primitive idea or basic idea about how to handle the data. Not here. Phasing, structure, we do not cover drug, but such application can be described in next semester. So you're interested in protein purification will be covered here. Protein and crystal as an extra. But protein crystal is very unique in terms of its demonstration of purity of your purification. If you have crystal, it demonstrates its purity of soluble protein or protein solution stock. Not
more pure, so more better crystal.
Usually pure, better. Without having pure, we cannot have packing. Even for NaCl, what is NaCl in common name?
Sodium chloride.
Sodium chloride in science, but
Salt.
table salt. Yes. We can take this as a way to purify protein or such a materials, table salt, even table sugar. Rather than having some mixture, once we have crystal itself are pure than aggregation or mixture. So always, even lysozyme, we have lysozyme as an enzyme. For commercial lysozyme, at least 3 times crystallized. So crystallization itself can be a purification step, high purity purification step. So 1 time, 2 times, 3 times means high purity, very high purity purification. But unlike salt, unlike sugar, without having proper purity already, we cannot proceed crystallization. Salt have strong tendency to be crystallized because they have ionic bond. But protein doesn't have such a strong interaction between its molecules. So without having some purity already in stock solution, we cannot proceed crystallization because impurity can hinder crystallization, unlike table salt or table sugar. By just lowering temperature, we have pure crystal out of mixture of NaCl and sugar. But even for them, to have very pure NaCl crystal or to have very pure sucrose crystal are not easy. Although common salt and common sugar are easily crystallized. Any crystallization is the most guaranteed purification step. Also a demonstration of purification quality. But our purpose is to have protein crystal is not directly for demonstration of purification, purity demonstration, but as a step to do crystallography. Okay. Here, what else do we have? Drug lead. What does that mean to you, Mohammed?
I think it will guide to the drug discovery, the structure information.
Here, these processes are common in whole or protein crystallography workflow, but this is unique to this drug discovery based on here VLS, virtual ligand screening, and SGD, structure guided drug discovery. Those are for drug discovery process for human and medicine. So from human gene, we have 30,000 protein expressed, and for disease, we have many proteins implicated in such disease-specific process. So if you have some small molecule like this, then we can design such this is lead, not the final. We can, if you have some affinity, you did some screening there. If you have some affinity between protein and drug lead, we can optimize such a drug based on protein structure. That's the part of drug discovery process. So this is only for drug discovery process targeted to some specific gene in human, but general, we will go through this process, gene to structure, based on genetic engineering or protein engineering. But target, and the word target there has something with the intention of developing drug for human. So drug target gene for drug discovery. So such connections or such importance have been attached to protein crystallography. That's the part of why protein crystallography are regarded as important science research in former days and even still now. But we will not deal with such a drug discovery step here. But this textbook was intended to help such researchers in drug protein structure-based drug discovery. So if we go in deeper in terms of workflow, number one, actually this step, cloning, expression, crystallization. You may be interested in cloning and expression and purification, right? But right now, Muhammad should do purification and crystallization. You didn't do any crystallization yet.
I said already.
You already said crystallization?
Yeah.
For what?
For the M cardinal.
M cardinal original.
Yes.
Okay, I see. Then you will proceed next step, collect the data soon,
But
coming month.
I have not checked either it has I got crystal or not. Just
When did you set up the crystal?
Friday.
Friday. Then it may take a week, or even 3 days or 4 days will be enough. So let's see. Friday, one day, almost 4 days or 3 days. Okay, let's check. If you have data, then either native single wavelength data or Ermer derivative data, they are kind of technical term. We will not cover this one, but phasing or molecular replacement, experimental phasing or molecular replacement, they are not topic for today, but later we will cover and skip this and model building. So based on the data collected here, we will build protein structure model. Then model deposit into PDB. So right now, you can skip these 2 columns because they are technical description for later chapters, but we will focus on mainstream, and model deposit means upload your PDB, your data, structure data into PDB. Deposit means make deposit into PDB. So that will be overall workflow and many details. You have corresponding chapters and if you are actually doing crystallographic analysis, as Muhammad will do, then you can go through particular chapters. But right now, after chapter 1, 2, you can study chapter 4 for most expression and purification issues. And we will cover soon. And let's go back to original question, opening question we had before, and let's summarize what we already did in terms of our understanding of crystallography, particularly biomolecular crystallography. Here, I'm thinking that we covered these two: why crystallography is important, and how crystallography research is possible. But if you have difficulty to answer these two, let's talk about and let's review a little bit on these two first. I just separate which from why and how. Why and how, we have no why, science issue, and no how, technology issue. But no which, I didn't see any word written like that, no which, but this is reserved for more technical terminology or more symbolic terminology like FHKL and low XYZ, electron density XYZ. So this is reserved for crystallographic vocabulary, but this is more general as you are interested in protein purification and gene cloning, and also some further application as a future career, as a science or engineer manager, or even funding agency. They are concerned about why important. But as a critical judge of some scientific or engineering project, how it can be done? Unless we know how we can do some particular project, we cannot fund such a project. So we should know how, we should know why, both. So we talked about protein data bank, and I just talked about something about drug discovery, need for discovery or workflow for this drug discovery. So why and how? Can you explain in your own vocabulary as your direct version? And
I think these words, these also have relation with these why, how, and which. So why has some relational aspect. Why crystallographic research is important. Why the crystallographic research specifically is important in comparison with other studies. So it has some relation with the development of the science, the structures, in structural biology, the development of structural biology. It has importance to develop the deep understanding, to understand the deep understanding and develop the structural biology in that sense. So it has importance in relation to other fields.
what other fields do you intend to apply?
I mean, it is like a comparison of crystallography with the other fields. Like if there are other fields also important, but crystallography has its own importance in understanding and developing the biology, and in promoting the deep understanding, the deep knowledge for applications in other biologies.
Simply, let me ask in this way: how can you use deep, the word deep, in this case, for crystallography?
Because of the molecular structure determination.
If I paraphrase what you told, so we have more detailed structural information than other technique like NMR. Is that what you mean?
No, not NMR. I mean, suppose we want to understand the functions
Mm.
of some process in biology,
Mm.
but we need deep understanding. That we can do by crystallography,
Mm.
by
What do you mean by deep understanding?
deep understanding of the molecular level understanding.
Molecular level. So we have molecular level of understanding even with NMR, even with neutron diffraction. But we need to be specific about deep. You said deep understanding, but deep, the word deep doesn't deliver any deeper meaning by itself. That is my problem to understand what you are trying to explain.
Broad, maybe.
Hmm? So, what is your own opinion about why and how and
Crystallographic research is important to develop and to understand the structure. So by
structure? Yeah.
So by understanding the structure, we get to know the function of the molecules.
Mm.
Just like by knowing the structure of antibody,
Mm.
we know how does it perform its function by attaching to the site, its site of action.
Mm.
So
So, if I interrupt at this moment, your word deep was translated into structure. Also, she gave us more specific and concrete example using antibody structure. Antibody is not direct target for drug, but antibody itself is part of disease and health problem. So if we know the structure of antibody, we can design vaccine, or we can develop even new antibody. So they can be useful technology. So rather than using deep, Saba provided us a concrete word, like antibody and structure. Structure of antibody simply. But what do you have in your mind?
I agree
Why?
with Muhammad and Sabah about the importance of crystallography. It's really important to detect the structure and function, and after that we can apply the structure and function of protein for other process. For example, crystallography, after we know structure, function, we know about the molecular protein, and apply for discovery the inhibitor of protein.
Inhibitor of protein
Yeah.
for
Yes,
drug discovery.
Yes,
Oh.
right. And it's really important for develop the pharmacist industry or something like this.
Mm.
Yeah. And how is how is it possible? I think based on the electron density and structure factor, it's really important data for how have us to know about the crystallographic, and it's really, I think it's one of technique we can apply.
Major technique we can apply.
Yeah.
In PDB statistics, 85%.
Yeah.
The other 15, almost NMR. So NMR, second major technology. Crystallography is first major technology. So dominant technology is crystallography for structural understanding of biology. Both of you provided concrete example, antibody and inhibitor for protein. So it's better to have some concrete example. And on top of that, I will add some more for why. As I told you, crystallography is first major technique for structural biology. And structure, understanding structure can be a first step to design biomolecules for our purposes, either as a antibody-based application or inhibitor design targeted to protein, okay? Or variety of researches of which information need protein structure, okay? So why important? Firstly, we need structure of proteins. Secondly, crystallography is major technique to have such a structure. But you can have such an answer in your own vocabulary, or you may have your own key concept about this. But it depends on your need. Also, how possible is, firstly, without having crystallography is not possible. So we have some bottleneck in terms of technique. NMR, it doesn't have such a bottleneck. NMR, molecule in solution, protein in solution. But for crystallography, we need a crystal out of protein solution. We have some extra step for protein crystallography. But once we have crystal, we have structure, we have diffraction image based on structure. So once we have such a diffraction experiment, we can determine electron density as a structure information. Okay? So anyway, you have now enough information. So we will jump into which. So remember FHKL and rho XYZ, and this one as your reminder, and we will describe. When we had this, you said picture. I said image. Picture, image are equivalent. Even photo. In early 60s, for first determination of hemoglobin and myoglobin, we didn't have camera. We didn't have digital camera. We have only film, X-ray film. At that time, we really had photo or picture of X-ray diffraction. But nowadays, even for human photographs, we are not taking film. We are taking digital image directly. Likewise, nowadays for X-ray crystallography, we do not use X-ray film. We are using a digital camera for X-ray, so-called CCD camera, or old days image plate detector. Okay. Anyway, we are using digital camera now, even for X-ray. Here, you can regard this as a digital camera for X-ray. Then let me ask this question: why we cannot see protein like this from this digital camera, unlike human photograph?
Because it's crystal, so it's transparent, so like the light passes through it. So that's why it cannot display its image.
That is good point. Transparent, not visible. So we cannot see proteins inside of crystal. Crystal itself, it doesn't have color usually, although we have some color crystal. Most times protein crystal not very colorful, it doesn't have very intense color. Even if it has a color, single color, because we have single protein, color is a single, not multiple color, red, green, blue, violet. violet crystal, red crystal, blue crystal. You have now blue protein, or
Blue. Purple.
out of blue protein solution, you may have purple or blue?
Purple.
Purple. Purple crystal. Not very many colors. Why we cannot see protein structure out of this digital camera? Muhammad.
I think because we are following the X-rays. The beam is X-ray,
Beam is X-ray.
so it will penetrate and pass through the crystal and will fall on the detector. So it's different from the digital camera.
In terms of recording signal, actually same mechanism in terms of imaging. What I'm saying is, if you have some signal here, those signals can be recorded as a pixel number. They are the same. For digital camera, we have color digital image, right? Red, green, blue. But here in X-ray, we have only intensity, no color. But each pixel contains some proportional counting, which corresponding to X-ray photon there. At this point, particular point, if it has more photon, X-ray photon, it has more spot or darker in terms of here, lower or whiter. This is darker or black. But still, we have some proportional counting, which corresponding to each photon here, like digital camera. Digital camera has a filter, color filter, which discriminate red photon, blue photon, green photon, RGB. So each photon proportional to each counting, so we have some composite image, colorful image. But here, we have same proportional counting as a digital recorder, but we cannot see a image like this. Do you know why? And you had some idea about wavelengths, didn't you?
That will be in the visible light, but this is not visible light, this is X-rays. So X-rays are have more frequency and less wavelength than the visible light. So it cannot be
Like as you did for deep. Firstly, but your contribution is greatly helpful. Muhammad, for previous sections, how and why, he used some broad term like deep. So the other two provided more concrete examples. Here, he said visible light, or if you translate light as a more technical term, visible ray. Beam. Light as a beam is ray. X-ray is a non-beam or a non-light. So like a deep understanding, he is saying visible. But beyond the word visible, we didn't get into some specific terms. Now, let's talk about in terms of wavelengths. What wavelengths do we have for visible light? I'm talking about ranges of such a wavelengths.
almost 400 to 700.
400 to 700 what?
Nanometer.
Oh, nice! Nanometer. Nanometer is 10 to the minus 9 meter, right? But what is nominal wavelengths for X-ray? Typical wavelengths for X-ray crystallography. For example, we need to talk about angstrom first. Wait, don't say anything yet. Today we started with English alphabet, then we extended a little bit in Greek alphabet, alpha and rho. Now we have another alphabet. What is this? Totimui.
Armstrong.
Oh, good. Then let me ask another question to Sabah alone. What is 1 angstrom in meter unit?
I don't know.
This is 0.1 nanometer. Firstly, this is not SI unit. Then, based on this, you can calculate this. (10 to minus 9 meter), 0.1 nanometer corresponds to which meter? Huh? You can help your friend.
I think you said 10 minus 10.
Oh, nice. You got help from your friend. We should help each other. So this much shorter than visible ranges. So think about microscope. When we are saying we are seeing something, either with naked eyes or with the help of some magnifying glasses or even with the help of microscope. What is the limit of microscope in terms of distinguishing some subject or object?
10 raised to power minus 6.
So with microscope, visible microscope, we have another microscope, electron microscope. With visible microscope, we cannot distinguish object less than this wavelength. This is the limit for distinction or limit of resolution. We cannot see almost half of wavelengths. We cannot distinguish them. With some sharp contrast, we may approach near to this wavelengths, but beyond this, we cannot see because visible light itself has interference at the order of this. This is level of 1,000 angstrom to almost, if we exaggerate this part, 10,000 angstrom. So visible light itself is much bigger in terms of length scale compared to angstrom. But what is the nominal or typical bond distance between carbon and carbon? I'm checking your basic knowledge in chemistry.
This bond?
Single bond.
Covalent bond. Yeah, single bond.
Double bond can be shorter and triple bond can be much shorter. Yeah. What is nominal? Huh? 1 point what? Very close. 1 point. But actually, single bond is close to 1.5,
Wow.
and triple bond can be 1.2. Okay. CH close to 1. Carbon to hydrogen, shorter than triple bond. Okay. So bond length is in the length dimension close to 1 angstrom. So because of this length scale of X-ray, so we can distinguish atomic distance. This is smaller than this. So most cases we can distinguish atomic arrangement within molecular structure because we have this. Theoretically, we can distinguish down to wavelengths, half wavelengths. Okay, but with visible light, we cannot see this. We cannot distinguish this. That's the reason why we need X-ray diffraction I didn't say X-ray alone, but X-ray diffraction to see molecular or atomic arrangement within the sample or within the crystal. Okay, so like a deep understanding. Muhammad said visible, but we should not stay at visible, the word visible. We should jump into angstrom, like concrete example of antibody or inhibitor molecule to target protein. We should introduce angstrom unit. Then we should put visible light in terms of angstrom unit. Then we can see huge difference between X-ray range and visible light range. Okay? You should have this idea. Also, the reason why we need X-ray diffraction or crystallography, with visible light, we have lower limit or minimum resolution to distinguish atoms in here sample, our crystal sample. So visible light, even very big magnification ratio, simply we cannot distinguish atomic arrangement because of limitation in its wavelengths. Even for electron microscope, we have down to 10 angstrom, several 10. So electron microscope has much better resolution than visible microscope, but still it has limitation. So X-ray, by the virtue of its wavelengths close to 1 angstrom, we can distinguish atomic arrangement. But unfortunately, unlike visible light, we do not have lens, magnifying lens. So we need X-ray diffraction or scattering. X-ray is not X-ray for chest, X-ray for X-ray CT, computerized tomography. This is direct imaging because we are examining human or animal tissues. In such a cases, we are looking at level of millimeter or sub-millimeter. We are not looking at micrometer or nanometer. It's okay to have direct imaging. As you said, transparent imaging. If you have less absorption through some tissues, we have some transparent region. If you have some blockage in terms of dense material inside of some tissues, we have darker. So opposite. Darker means transparent, and white means some bones blocking X-ray. So we have chest photograph, X-ray photograph. But unlike chest photograph or computerized tomography, we should see ranges like this. So we cannot rely on direct imaging. So we should use indirect imaging of X-ray crystallography. We are not doing direct imaging, unlike X-ray CT, unlike X-ray chest photograph. We are doing indirect imaging. That's the point described here in this image. But still, let me give you some idea using direct and indirect. You should be careful about listening to my word. Here, this is direct image of, not of crystal, but of structure factor. Crystal, if this is direct imaging of crystal, we should convert directly to our understanding of structure. But this is not simply possible because of this great difference in length scale. So let me repeat this again. You were trapped in my pitfall designed. What I'll say firstly, what Saba tried to say this photo is direct imaging of crystal. But this is not true. I'm sorry. But this photo, direct image of structure factor of the crystal. Not direct image of crystal, but structure factor of crystal. Kind of exaggeration of articulation. But why did I use direct here? Because this is physical event, not computational event. From here to there, if we have crystal sample here, really we have some spot there. Like SDS gel learning, if we have protein spot corresponding to some molecular weight, we should have spot. Even for DNA gel learning, agar gel learning, if we have some DNA fragment, we should have DNA fragment spot there. Likewise, if we have crystal here, we should have some regular spot there on top of digital camera for X-ray. So I said direct imaging of something, but something shouldn't be crystal itself. Okay, that is a secret. But if you are saying this is indirect imaging of crystal sample, okay, fine. This is indirect imaging of sample, but still direct imaging of sample. Sample is sample, but we can regard this sample in two ways. One is as a real space crystal, sample in real space. The other way is sample in reciprocal space. Single sample can be understood in two ways. Sample in real space, sample in reciprocal space. Both are possible. That's the secret of crystallography. You should develop such a mentality. Single event in two ways. So still we have direct real-time imaging of sample rotated. Usually we are rotating crystal sample in 180 degrees. I'll explain later, not today, but later days, about why we do not need 360 degrees. Because of symmetry, we just need half of 360. Okay. Anyway, this is direct imaging of sample, but not in crystal of real space, but in sample structure factor in reciprocal space. Then, if we regard as an indirect image of crystal, we need Fourier transform back to electron density. If we regard this as a indirect image of, this is a little bit funny here. These are equivalent. These are in contouring imaging of protein sample, and this is what protein? What presentation is this, Muhammad? In this morning, I worked with you for Pymol presentation. And how do you say this? What is this? Here we have two different presentations of protein structure. I gave you a difficult answer to this one. This is electron density contouring. I said contouring. This is kind of technical presentation in protein crystallography. Contour, like a contour in Google Map or Earthly Map. We have contouring in crystallography, kind of leveling, contouring. This is contouring presentation of protein structure, and this is another presentation, not in contouring. What presentation do we have here? We have some red and some cyan, but what's in red and what's in cyan? Oh, nice! Helix in red, and what else do we have in cyan?
Ribbon. Ribbon?
Beta sheet. It's a flat, or a flat. The others are like coil in white. Okay, so 3 different colors are used for secondary structure: helix, beta sheet, and coil. Okay, so-called pitch presentation. Muhammad, I used such a terminology in this morning, and in PyMore, in molecular display program, we have, what is this? Ribbon. So, ribbon, girls like ribbon. So, gentleman doesn't know anything about ribbon. So, this is ribbon presentation of protein structure. This is contouring presentation of protein structure. Both are valid presentation for crystallography. But ribbon structures are more general, not only specific to crystallography, but also for drug discovery design and any kind of, even structure paper contains, all structure paper contains like this. Even biochemistry textbook contain lots of presentation like this. It's called ribbon presentation. Above is electron density contouring presentation. And both are protein structure. Here, Fourier transform give you directly this presentation, then we should convert this presentation down to here. Because this presentation doesn't contain any information of alpha, beta, coil, but with this kind of coloring, it's much comprehensive or understandable, right? Oh. So when you read textbook, develop your own question like this, and if you have difficulty to understand material, bring such a question to the classroom or coursework. Okay. Here, here used Fourier transform. Fourier transform was given there as a way to transform data, this image to another, this structure. So structure information in electron density, structural information in ribbon presentation. Both are equivalent, but initially we will get this. But from this to that, we should have another assignment in terms of atomic assignment. Here, we do not distinguish atoms yet. We just know contouring. Based on electron density contouring, we should assign atoms. Based on amino acid sequence or our pitting, we should assign some atoms. Then we have amino acid, corresponding amino acid, and we have this kind of connection diagram. See? So, another complexity. Here, two things are coupled. What is this contouring in blue? I'm checking your understanding. I'm trying to hear about the word to describe this one.
Electron density.
Oh, yes. Electron density. Contouring. Electron density. Contouring of electron density. And here, this is not ribbon, this is stick. Another presentation. Ribbon and stick. For molecular presentation, contouring for electron density presentation. All of them are way to present protein structure. All are valid, but each one has different information amount. Contouring itself, it doesn't have atomic information, but ribbon and stick have atomic information at different level. Ribbon at secondary structural level with different colors. You can assign different colors as you need. Not necessarily red for helix, you can assign different color to helix. But you need to distinguish helix from sheet and coil, then such a structural element will be very visible. But here we are looking at atomic level, and here yellow one may be a, let me ask this again. Look at this. This is paneling, and yellow one, you can guess which atom we have in yellow.
Carbon.
Oh, nice! Or have correct answer. Carbon in yellow. Then what this will be in peptide bond? Oxygen, correct. And what is in blue?
Nitrogen.
Yes.
Amide.
Yes. So peptide and amide bond. So we can distinguish each atom by different color, but all are in stick. We do not have ball. Even we have ball and stick model in chemistry, but in protein crystallography, we rarely use ball, because if we have ball there, our picture will be so clouded. In chemistry, we rarely use electron density, but in protein crystallography, we have more ambiguity in terms of density. Always we use electron density overlapped with stick. Okay, in such a way we can check accuracy in our assignment of each residue assignment. Okay, so based on resolution, I will not explain the definition of resolution, but just take this number: 1.2 angstrom, 2 angstrom, 3 angstrom. High resolution, low resolution, detailed one, rough one, in terms of grid, watching grid. Then we have better resolution here as a donut-like electron density in paneling, but low resolution like 3 angstrom, we have some ambiguity in terms of distinguishing donut, but still close to donut, flat one. So depending on the quality of data or diffracting power of sample, we have varying degree of accuracy or precision in terms of seeing molecular details. So lower resolution, higher resolution, worse overlapping and better overlapping. We do not have any ambiguity in terms of distinguishing penicillin there, but we have some ambiguity in terms of finding or assigning different amino acid residue type. You should develop such an idea. Anyway, here already we have non-SI unit angstrom, 10 to the minus 8 centimeter or 10 to the minus 10 meter were given there. So you will not have any difficulty to find definition. And here, CC single bond 1.4 or 5.4. They add one more digit. Okay. Carbon itself has an atomic radius of 1.77 angstrom. Okay. So accuracy versus detail. And we started with two symbols, two different kinds of symbols. FHKL, pector. Pector became F there. F, pector. This is my guess. Such a thing was not explained in any textbook, but based on structure pector, not S, because S is also used in physics. S stands for scattering, so someone would like to distinguish between structure and scattering, so rather than SHKL, SHKL is a vector description of HKL. So they reserved pector F as a symbol for structure pector. That is my guess, which is not explained in the textbook, but you should take F as a pector for your own memory. Pector, F. Pector based on HKL coordinate. Density based on XYZ coordinate. remember what we did for Fourier transform. Time domain and reciprocal time domain frequency. Frequency also f. Like frequency, we have factor f. Okay, this is only for your memory. Factor, frequency, domain, HKL. Such a connection you should develop in your vocabulary in crystallography. X, Y, Z, electron density and structure, and ribbon presentation or stick presentation depending on accuracy or details. So we have good accuracy or details here, paneling and electron density overlapped with stick presentation of your protein structure, and FHKL not to be very precise, but for our own rough understanding, these are like a direct image of diffraction pattern or direct image of reciprocal space. Here, but two dimension, H, K, L, this line. Then,
2D.
yeah.
3D.
Here, this is not really 3D. X, Y, X, Y, and likewise H, K. Okay, L this side, Z this side. We have some 1-to-1 correspondence in terms of matching. So this kind of three-dimensional space in reciprocal space can be converted to another kind of three-dimensional space, vice versa. Here, inverse Fourier transform transforms F into electron density, but by Fourier transform, we can transform electron density to FHKL. Both are equivalent. So if you have this information, we can get this information. But if you have this information, we can have this information through the computation. I'm talking about computational aspect of protein crystallography. That's all you need. No more, nothing else. But we just develop some concrete understanding based on this main idea. No more main idea. That's all main idea. That's all main vocabulary. FHKL and electron density at XYZ.
What is we?
Hmm?
We.
Ah, nice question. What is we? We stands for we something. We in Ångström to 3 dimension or meter triple 10 to the minus 30 meter squared. We in such a dimension.
Space.
Speed.
Something in space, but what's in space?
Speed.
It's not speed. Speed has time.
Speed.
Ångström square, Ångström in length scale, Ångström square in area, and Ångström triple in V. Okay, you know answer now.
Volume.
Yes, volume. Volume. This is volume of something, but you will find what is something later. Very good question. I didn't explain V, but you have interest on V. You will find the definition of V later. Any HKL, this is a little bit tricky. The author of this textbook were not very exact in terms of rigor or precision of his mathematics. So if I'm correct, here HKL, their HKL are not the same HKL, even though he used the same symbol. In terms of summation symbol, we should use different, let's say, capital HKL or H minimum H maximum there. Something wrong here. Even there, something wrong here. I explain later. But overall, you should not be stuck in the details of this, even this. You should know the relation between FHKL, rho XYZ through Fourier transform. Fourier transform and inverse Fourier transform connect these two spaces. That's the main idea you should have, you should develop.
In reciprocal space and in real space.
Mm.
In reciprocal space and in real space.
In reciprocal space and in real space. Yes. Reciprocal space in angstrom, I made a mistake. Real space in angstrom unit. Reciprocal space in inverse angstrom unit. Okay? But imagine angstrom in length scale, actually if we say CC triple bond is around 1.4. If this is 1.4, actually this is 1.5. Actually, not double bond, not single bond. It's between single and double, and 1.4 something. If this is 1.0 angstrom like this, so 1 angstrom. But in real physical dimension, it corresponds to almost 3 centimeters. So this is not really in scale. You should notify this kind of scaling problem too. If we draw this in real dimension of angstrom, we cannot see this. Why? This is smaller than visible light. So because of that, we magnify this a lot. Angstrom to centimeter scale. We scaled it for our own presentation. Likewise, angstrom inverse is not really in physical dimension, kind of imaginary dimension. So here we have some direct scaling, but here we have some arbitrary scaling. Likewise, here, this image, if I'm honest or technical, if I'm trying to be technically correct, this two-dimensional image are not directly related this, but they are related. They are still related. But for your own level, for your understanding at current level, you can regard they are almost identical. This one is identical to this one only when we have crystal in special alignment. Here, if you look at this, crystals are slightly tilted. As an experimenter, if you are trying to put crystal there, you cannot perfectly align them. They are susceptible to handling. So either you can put this way or this way and this way. So crystal cannot be perfectly aligned with respect to its sample axis. So this one, to be more technically precise, this one can be distorted image of reciprocal image of reciprocal space. This is kind of regularized one. X, this, not X, X, Y, then H, K. Likewise, if you have some alignment, in precise alignment, we can have H, K. But in reality, that cannot be achieved because of arbitrariness here in terms of crystal mounting or crystal handling. Also, we have other issues of scaling due to the geometrical arrangement, detector geometric arrangement, also rotational aspect of sample. So we do not have one-on correspondence between this image, diffraction image, versus reciprocal image like this. But still, they are related. They are related. So you can regard this image as a regular one and previous image as a distorted image. So diffraction image can be regarded as a distorted image of reciprocal space. Okay. Also, we have some scaling issue in terms of choice of this. So on top of crystallographic problem, vocabulary problem or technical problem, we have some presentation problem. Like our choice of ribbon stick contouring, we have some presentation issue in terms of displaying reciprocal imaging of our crystal sample. It can be in regular, it can be in distorted view. This is regular view, previous one is distorted view. But both are presentation of reciprocal space sample. Okay? Do you have an idea about cosine and sine here? Otherwise, we will skip this. Yes, FHKL equal to FHKL exponential alpha HKL.
Fourier transform.
It's a part of Fourier transform, but as a single symbol. Let me explain.
Structure factor.
So simply to help you, I'll explain for myself. FHKL, as you said, structure factor HKL. Okay, but I put F in boldface to be technically correct. But here he put F in italic with alpha HKL. So if I expand boldface FHKL into some terms like this, we can have two versions. FHKL in italic multiplied by exponential alpha HKL. Here, alpha HKL, we can use another Greek alphabet like a pi. Although textbook alpha was used. Or pi. What is phase angle, so-called phase angle. Was a symbol for angle, and here alpha HKL was used as a phase angle, and structure factor HKL can be decomposed into two parts. Structure factor amplitude as a italic FHKL multiplied by exponential over phase angle. That was explained already in Wikipedia. Wikipedia. Structure factor. Structure factor. Let me skip. I cannot find it. Prompt 3.
In previous slide, first slide.
First, right?
Before this, yeah. Before this.
Before?
Yeah, this is structure factor.
That's the one I put. No, from Wiki.
Not from
I put for myself. Anyway, let me come back here. Simply by mathematics, exponential can be decomposed into. I missed one thing there. I myself made mistake. Here, I missed imaginary number I. If you have I there, you can decompose exponential term into two terms, cosine term plus I sine term. I missed I by myself. But here in the textbook, they made a mistake in here, not minus but plus, and we should have parentheses here. So exponential plus I alpha, not minus e pi I alpha. E pi is 360 degree in degree. E pi 2 pi is for radian, and alpha should be also in radian, not in terms of turn like this. But here, the reason why I put comment like this, to help you not to be deceived by textbook. I said deceived. Here in the textbook, it has some mistakes. If you have some higher level understanding, you will not be trapped by such pitfalls in terminology. Here, firstly, if you need to look at this, you have question about V, then you may have question about HKL, as I told you. Also, you may have question about extra alpha, so I put alpha here. But I missed I here, so please ignore this. Then, if we put boldface FHKL as a symbol for structure vector, then we can be able to decompose into two terms, FHKL and angular term in sine and cosine. That's what I'm trying to help you. Here, F and alpha HKL should be coupled like this, then multiplied by exponential minus 2 pi I multiplied by Hx plus Ky plus Lz. This term, I like to explain this term here today, but this has some significance in terms of interference. Here, in Fourier transform summation, HKL multiplied by XYZ. Accordingly, no cross term. H multiplied by X, K multiplied by Y, L multiplied by Z. So, remember this. In frequency, we had 1 over T, and or simply T, then Ft equals to 1. Okay? Likewise, if we multiply HKL part by XYZ part, we have some interference issue if they are integral. Multiplication of these are integral. They are in terms of a multiple of 2 pi integral. Then this will be 1. So this will be in constructive interference. Otherwise, we will be in destructive interference. So this summation has part of interfering contribution. Okay, that's the nature of Fourier transform. So I'd like to give you some idea of interfering nature of Fourier transform, also interfering nature of diffraction as a physical event. Through such an interference, we can multiply our signal. Like a PCL, we need to amplify something. Like PCL, the way we can amplify diffraction or scattering signal is through the periodicity of sample. What is crystal? Crystal is made of unit cell which are multiplied in great ways, many ways, over billion copies of identical packing unit. Through such amplification, we can see constructive interference like this reciprocal space observed in digital camera of X-ray. That's the diffraction process. Okay. But on top of that, we have some errors there. My error, textbook error, and you should not be prone to errors. You should have higher level of understanding, not technical level of understanding. And all figures are from textbook, so you can review your textbook, and you should be a judge of such a technical error. I'll show you another error or another long description which was given in figure 1. I took this equation from figure 1, and in the figure legend, the author talked about its accuracy, but as I put numbers, it wasn't very accurate. I took this formula and I put year like this, year, number of available deposit at particular year. I put 2 years, 99 and 2004. I got these two, but they are in the order of this, not in this order. One order lower, the other one order lower. So 0.19 may not be very correct in this application. So I had some doubt about this, and I tried to find better coefficient there. This is what I did as a correction. Anyway, read the footnote or figure legend. The author talked about its accuracy, but I found by putting real numbers there, not very accurate. So I made some correction like this. Based on year 2004 and year 2005, here, 30,000. So by putting 30,000 as an n and taking log at both sides, and I tried to find correct number rather than 0.19 by dividing year minus 1960 there. So I got new number rather than this number, 0.23. By putting 0.23, I got this and this, which is near to this and this. That's what I did for the revision of figure 1. But if I put this year 2015, year 2015, we have this report. But by putting year 2015, it predicts this much, 4 times different. So here, quite good in terms of its prediction by this correction, but not quite good beyond year 2015. So year 2015, the author might not check it for himself because he didn't write this time. But this and this are well in terms of publication year of this textbook, but he didn't check in this way, so not very correct. So through these 2 examples, this one and previous one, I'm advising you to check material for yourself. When you are really concerned about accuracy related to your own outcomes on your own success. If this is only about coursework, it's okay to ignore whole kind of checking. But if this is not just coursework, but for your thesis work, it's better to check its accuracy. Even equation used for protocol, some protocol may have wrong equation, which are not valid beyond printed table. So you need to check its accuracy, even for equation or even for table numbers like this. Okay? So what I'm advising you, particularly Muhammad, don't believe textbook. Don't believe even published one. To be really successful in your project, you need check when you are concerned about its accuracy, when you should depend on the accuracy. Then we need check for ourselves. Then you will not be failed. Your success will be better guaranteed, not by others, but by yourself. That is my take-home message. Don't trust others. Trust your own intelligence. Even if you are not a specialist at new field, you need to take numbers and you should plug the numbers into equation and let's see. Also here, you ask question about V. Likewise, you can ask about alpha there. I would try to give you explanation of alpha, but partly I made my own mistake. I introduced a new mistake there. Okay, so that is human problem. Humans are prone to mistakes, but scientists should be free from mistake by careful practice. Question or filling this sheet, or you can start filling the question sheet and you can ask questions during this session. So as a second, after break, I covered which crystallographic data are needed. You may have some idea and you can use some symbolic presentation or you can use some English vocabulary. So you can fill the third part for the question.